生命起源于何处？又将归于哪里？记录、察觉、唤醒，小小的细胞中隐藏着未知的秘密。当生命科学成为人的延伸，我们看见彼此更细微、更光亮。西湖大学生命科学学院致力于研究生命现象、疾病机理，探索发现生物医药的新技术和疾病诊治的新方法。在这里，我们是无畏的探路者，从萌芽一个想法，到开始一次试验，大胆猜想，小心求证，一场场冒险，一遍遍反思，不断追问生命现象的本质。在这里，我们是进取的创新者，平等包容，学科交叉。无论是讲席教授，还是青年科学家、学生、行政人员，我们彼此尊重，自由思辨。在这里，我们是梦想的守护者，鼓励大家去提问，去想象，保持对科学最纯粹的兴趣与好奇。探索未知的科研世界，每一次思考都是对生命科学边界的求索，每一次验证都是对人类生命健康的守护。敬畏、探寻、发现，我们将共同创造更加美好的未来。
尊敬的各位来宾，本次会议即将开始，请您尽快就座，并将手机等通讯设备关闭或置于静音状态。谢谢您的合作。
Ladies and gentlemen, the event will begin shortly. Can we please ask for you to kindly take your seats? We would like to remind you again to switch off all mobile phones and put electronic devices to silent mode. Thank you for your cooperation.
Okay, we'll get started. Good morning, dear speakers, colleagues, and trainees. Welcome to the 2021We Forum, We Academic Forum. The theme of the forum is infection and immunity. So the stated purpose of the forum is actually to celebrate the new opening of our new campus, Yungu campus. But as expected, everything is behind schedule, so we're not ready to hold the forum in the new campus. We have to hold it here. But I think the spirit is correct, so we are really happy on this occasion that our new campus will be soon ready for use. So I think speaking as a non-immunologist, I can speak for many of you in the audience that immunology is the hardest subject to learn. In fact, when I was looking for faculty positions in the late 90s. If you look at the ads for faculty position for immunology, oftentimes the deadlines were extended because of lack of qualified applicants. I think the recent years, the success of cancer immunology has really brought a renaissance back to immunology. Now it's one of the hottest areas in research. And also with the current ongoing COVID-19 pandemic, it really reminded us there's a lot of gaps in our knowledge in immunology and infection. So I think this forum is very timely. So of course, the most important part of the meeting is the lineup of speakers. So the organizing committee, I think, except myself, everybody else has done a lot of work. Dang Shengli, Chang Xing, Xu Heping, He Danyang, Liu Zixu, and Zhou Ting are the responsible parties who bring together this outstanding lineup of speakers. We'll have a combination of online and in-person presentations. So they cover a wide range of topics from plant immunology to the interface of neural and immune systems, and of course to cancer immunology and also SARS-CoV-2 mechanisms. So I think this meeting is also sponsored by the Westlake University and the Westlake Laboratory of Life Sciences, and also the World Young Scientists Summit as part of their meeting series, and also is sponsored by Sihu, I think it's Lucy Micro. It's a startup company funded by our own Tao Liang. So let's thank Liang for supporting this meeting. And also finally, I'd like to thank our administrative team led by Kaina. I think they were testing, and also our team from the outside to really run this meeting both online and offline. They were testing the setups till I think 3 o'clock in the morning. So let's give them a round of applause for their dedication. Finally, I want to say to the meeting, the main purpose of the meeting is, of course, for the trainees. So I hope the trainees will get inspirations from our successful speakers and scientists and find your own interest and niche, and eventually you'll be the future in life sciences that will help hopefully bring the pandemic to the end and also solve major problems in life sciences. I wish I hope the meeting will be a resounding success, and thank you very much for coming. Oh, I now turn the podium over to Zhou Ting, who will chair the first session.
Good morning, everyone. My name is Ting Zhou, and I am a faculty member of Westlake University School of Life Science. So I will be the session host for the morning first session. So we're very happy to have Dr. Akiko Iwasaki as our first opening keynote speaker. Akiko is a professor of Yale University Department of Immunobiology and also Department of Molecular, Cellular, Developmental Biology. Her lab studies innate and adaptive immune response to a variety of viruses and how this virus infection leads to disease consequence. So her lab made a lot of contributions to this field, including the TLR7, TLR9 signaling in tissue-specific dendritic cells, and the prime and pool vaccine strategy, as well as endogenous map in human genome. So for these early accomplishments, she was recognized as Howard Hughes Medical Investigator since 2014. She was also elected to National Academy of Science in 2018, National Academy of Medicine in 2019, American Academy of Microbiology in 2020, and American Academy of Arts and Science in 2021. And more recently, since the start of this pandemic, as a virologist and also immunologist, Akiko and her lab quickly turned to be a major task force working at the forefront of COVID-19 research. Together with the clinical team, her lab published a lot of important papers addressing key questions in SARS-CoV-2 study. I believe her talk will cover part of that. And outside the lab, she also very active in the social media like Twitter. You can follow her Twitter, Virus Immunity. And there she will be very patiently to explain and communicating COVID-19 facts and also her scientific findings with the public. So she's also quoted by a lot of TV reporters and newspapers as for her expertise insights. And she's also considered one of the top 50 experts to be trusted during this pandemic. And also not only as a good scientist, she's also very great and supporting mentor for the trainees. She's also well known for her advocacy for the women and underrepresented minority groups in the science and medicine field. In today's talk, Akiko, I think she will talk about the immune response to SARS-CoV-2. With that, let's welcome Akiko to give her talk.
Thank you. Thank you very much. Can you hear me? Yes? Okay, I'm going to share my screen. All right, so today, as Ting mentioned, I would like to talk about our recent work on immune responses to SARS-CoV-2. One of the key insights that the immunology community is getting is the sheer variation in COVID-19 disease outcomes. This has to do with, of course, the host, such as the age of the host, as well as the sex, as well as pregnancy status and other conditions that might accompany a person's immune response and infection. And we, together with a number of collaborators, have delve into the immune responses to SARS-CoV-2, looking at patients that are in our hospital with moderate, severe to lethal COVID, and dissecting the different types of immune responses that happen. And our insights include, for instance, the severe COVID patients having larger amount of viral titer, and that are having misfiring of the immune responses that correlate with the immunopathology that we see in COVID patients. We also demonstrated that the timing of the immune response is important, that the neutralizing antibodies that develop early in the immune response is helpful, while those that develop later are not beneficial. We've also done a lot of work on looking at autoimmune responses that happen during COVID, as well as the neuro-COVID and the impact of saliva viral load on disease outcomes. We also turned our attention to looking at differences in sex and immunity. And this in particular, we focused on early phase of immune responses in the patients that are hospitalized. And we found, for instance, that early phase immune response, particularly T cell activation, is elevated in female patients, while those in the male patients were reduced, and that correlated with the worsening of the outcomes. And we've also looked at the impact of COVID and pregnancy, and have reported one severe case of infection that resulted in infection of the placental cells, as well as respiratory tract infection resulting in impact in the placenta. So today I'd like to basically talk about three separate studies. One study has to do with how different mutations that accumulate in the variants impact immunity induced by the vaccines. The second story is a mouse model of COVID-19 that enables us to dissect the different effectors involved in clearance of the pathogen. And finally, I'd like to talk about our ongoing work on long COVID pathogenesis. The first study I'd like to share with you was led by Dr. Carolina Lucas. She is a very talented postdoctoral fellow in the lab. Together with Dr. Chantal Vogels, she is a brilliant postdoc in Nathan Grubal's lab who does viral surveillance. And Dr. Insi Ildirim, who is a pediatric infectious doctor who collaborated with us on the story that I'm about to tell you, along with a number of collaborators. So the study design was to enroll healthcare workers who had either been previously infected, that are indicated in the blue, and previously uninfected, in the purple people. And what we did was we collected blood at baseline prior to vaccination and 7 days and 28 days post first dose of mRNA vaccine, and 7, 28 days or 70 days post second dose of the mRNA vaccine, and compared their antibody and T cell responses. What we showed is that the mRNA vaccines are generally very good at inducing high levels of antibody. So on the top is the anti-spike IgG. The middle panels are the S1 domain of the spike, and the bottom is anti-RBD, the receptor binding domain of the spike. And you can see that at baseline, if you look at the right panel, the vaccinated, previously infected people had some levels of IgG to begin with. But after one dose of mRNA vaccine, which is the time point 1, you start to see already a huge elevation in their antibody titers. And then they maintain very high levels of antibody. Whereas previously uninfected people also get there with 2 doses of mRNA vaccine. Of course, they start out with no antibody to begin with, but this quickly catches up to those who have been previously infected and vaccinated. And when we examine the ability of the mRNA vaccines to elicit neutralizing antibodies, so that this is using authentic virus of the original SARS-CoV-2 lineage, what we see is that, again, if you look on the right, the patients, the participants who were previously infected are indicated by blue dots, and those who are not are indicated by purple dots. And you see that there is some baseline levels of neutralizing antibodies in the previously infected participants, but this levels goes really high, even just after 1 dose of mRNA vaccine, and maintained very highly. Whereas those participants who had not been exposed to the virus are able to catch up after the second dose. So this is all good, that the mRNA vaccines are eliciting robust neutralizing antibodies. We also demonstrated that the T cell immunity is robustly induced by the 2 doses of mRNA vaccines. This is looking at 28 days post second dose of mRNA vaccine. On the top are the CD4 T cells, and on the bottom are the CD8 T cells. And shaded in purple are the previously uninfected individuals, and shaded in blue are the previously infected. And what I've done is to just sort of highlight the activated T cells, which are HLA-DR positive and CD38 positive. And those types of T cells are observed very readily when we stimulate the T cells with the spike protein from the original lineage A, but also when we tested against a P1 variant of concern. We also saw a very robust CD4 and CD8 T cell response. There was some reduction in the CD8 T cell response to the spike of the P1 variant. However, this definitely remains above background. Next, we turned our attention to the different types of variants that are circulating, or that have circulated in our community. And on the panel A on the left, what I'm showing you is the 16 different isolates that Carolina was able to purify out of the nasopharyngeal swabs of people who are infected in the community, and genotyped by Chantal. And what you see is that there are these gradients of variants that have different amino acid mutations that accumulate in the spike region, all within the N-terminal domain, the RBD, the RBM, and the furin cleavage site. And on the right, what I'm indicating is the neutralization titer from these various individuals that were vaccinated twice with mRNA vaccines. So against the ancestral strain, I told you that vast majority of people, except for 2 people, develop high levels of neutralizing antibodies. And if you go down the list of variants, it really doesn't make a big difference until you get to about the Delta variants in the greens. You start to see a significant drop in the neutralizing antibody capacity induced by the vaccines. And this coincides with the mutations such as the L452R mutation, which single-handedly has the most impact on escape from neutralization antibody, which is shown in the right. And you go down this list even further, you see the epsilon and eta and kappa, you start to see some more decline in the neutralization titer. But when you get down to the gamma and the beta variants, they're the most evasive or have the highest resistance against the neutralizing antibodies. And on the right, what I'm showing you is the impact of each amino acid mutations within the spike region that have the capacity to escape this neutralization. What's rather striking about the finding is that if you look at the inter-individual variation in the neutralizing antibody response against the variants that are listed across the screen here, you see that people who develop very high levels of neutralizing antibody against the ancestral variant strain have the sort of a capacity to have cross-reactive antibody responses all the way across to the beta variant. Whereas people who started out with lower levels, they tend to drop off in their ability to neutralize variants much quicker. And of course, the 2 people that I mentioned somehow were unable to develop neutralizing antibodies, even though they had quite a good levels of ELISA detectable anti-spike and RBD antibodies. So if you separate these participants into those who are vaccinated on the bottom, they're all vaccinated, those who are previously infected on the bottom versus previously uninfected on the top. What you see is that those who had never been exposed to the virus but have been immunized twice with mRNA vaccine are able to control, of course, the ancestral variant strain, but their ability to neutralize different variants demonstrates rapid decline in their ability to do so with variants that are indicated here. Whereas those who had prior infection and then were vaccinated are able to maintain cross-reactive antibodies, across many, many different variants. So this isn't a big surprise because prior infection does confer another exposure to the antigen. So it's almost like having a priming dose prior to getting the vaccines. So this study demonstrated that the mRNA vaccines do elicit robust antibody, neutralizing antibodies and T cell response against the spike. And previously infected people are able to do so even better than previously unexposed individuals. And that there are distinct mutations in the variant that confer different degrees of escape from the neutralizing antibodies. And certain mutations do appear to synergize to escape the neutralizing titer, such as the Delta H69, V70 combined with the E44K mutation. And finally, we believe that these data sort of illustrates the continuous need to conduct viral genome surveillance across the globe so that we can anticipate what type of variants might be arising that may dominate the different population. Currently, the Delta is dominating the world. However, there may be new variants that arise in the future that might require variant-specific vaccines. So now that we've learned much about these vaccine-induced neutralizing antibodies, we wanted to understand other components of adaptive immunity in conferring resistance against the virus. And this was enabled by a strategy that Dr. Ben Israelo a infectious disease fellow in the lab, developed in the very beginning of the pandemic, which is to develop a flexible mouse model of COVID-19. This question really was elicited by a number of different studies looking at the correlate of protection against SARS-CoV-2 infection. On the left is a Nature Medicine systematic review of different vaccines or infection that generates different amounts of neutralizing antibodies that have a very nice predictive ability to look for protective efficacy. And it's not only the neutralizing antibodies that are important for protection, the non-neutralizing viral binding antibody can also engage the Fc receptor on immune cells to control the virus. And finally, T cells are obviously important for generating antibody responses, as well as acting as effector cells to control infected cells. So there are some really nice evidence out there from real world data that look at these correlations, but we wanted to understand what are the relative importance of these effector functions. So Ben took the approach first to look at the role of B and T cells in clearance of the primary SARS-CoV-2 infection. To do this, he utilized a model that he created, which is to transduce mice of any genetic background with AAV encoding human ACE2. And this enables any mouse of any genetic background to become susceptible to SARS-CoV-2, any human isolates that we have in the lab. And then he can measure disease and viral load in different tissues. So on the bottom of what I'm indicating is the infectious viral particle per mil of within the lung after infection of the wild type animals that are transduced with human ACE2. And you see that there's a very rapid and complete clearance of the virus within 7 days. However, if you look at the MUMT mice in which there are no B cells, these mice are also able to completely clear the virus within 14 days. And at 7 days, there are some low levels of virus detectable in these animals, indicating that B cells are not necessary to control primary infection. However, when Ben took rag knockout animals, these are mice without any T or B cells, these animals were unable to control the primary infection even at 14 days post-infection, and they actually developed chronic infection with SARS-CoV-2. This illustrated the necessity of T and B cells, the adaptive immunity, for controlling primary infection. He then turned his attention to the importance of T cell subsets, CD4s and CD8s, in controlling primary infection. To do this, he again took advantage of the human ACE2 transduction system. And on the top, what he did was he transduced wild-type animals with human ACE2. And then he depleted either CD8, CD4, or CD8 and CD4 T cells and measured viral titer in the lung at 14 days post-infection. And what he saw is that depletion of CD4s or CD8s alone had no impact on the clearance of the virus, indicating that these subsets are not required for clearance. However, when he depleted both of these subsets, then he started to see some animals still harboring virus at 14 days, indicating that you need T cells for complete clearance of the virus. It turns out that in these animals that are depleted of CD8s and CD4s, there was also reduction in antibody responses because of the necessity of CD4 T cells to elicit, provide help for B cell to produce antibodies. And on the bottom, he did the same exact experiment in the MUMT animals that don't have any B cells. And in this setting, what he saw was that depletion of CD8s or CD4s had basically dramatic impact on the ability of these mice to control the virus, indicating that in the absence of antibodies, CD4s and CD8 T cells play a major role in controlling the virus. And when he depleted both CD8s and CD4s, he converted these mu-MT mice into a rag knockout animal setting in which there are no adaptive immunity present to control the virus. So what about the role of B cells and T cells in protection against reinfection or vaccine-mediated protection? To get at the reinfection question, first, Ben used an adaptive transfer system to see if antibodies or T cells are sufficient to confer protection against infection. To do this, Ben took serum, which contains antibodies, or T cells from these animals that were already immune to the SARS-CoV-2 virus and transferred them separately into rag knockout mice that are transduced with AAV encoding human ACE2. He challenged these animals with SARS-CoV-2 and measured virus in 7 days. And what he found is that the T cell transferred animals were able to significantly reduce the viral load in the lung within 7 days, and some of them completely clear the virus. So indicating T cells are partially responsible for clearance of the virus. However, the serum transferred animals, none of them had any detectable virus within 7 days, indicating the ability of the serum antibody to completely control virus infection. Then Ben took advantage of the mRNA vaccine to create a situation in which animals have varying degrees of antibodies. To do this, Ben took either 1 microgram, 0.3, 0.1, 0.03, 0.01 microgram of mRNA vaccine, gave it to a K18 mice. These are animals that are transgenic for human ACE2. And then he measured antiviral antibodies in these animals. There was this really beautiful dose-dependent response of antibodies generated by different doses of mRNA vaccine, 1 microgram being the best in the blue, followed by 0.3 in the red, 0.1, and so on. And then at 0.01 microgram, there was no detectable antibodies. He took these animals, he challenged them with SARS-CoV-2 and monitored for disease by weight loss or survival. And what he saw was that the mice that received either 1 or 0.3 microgram of mRNA vaccine were completely protected from disease or death, whereas animals that received 0.1 microgram, there were about half of the animals died, and there was disease as observed by weight loss. And any animals getting less than that amount, 0.03 or 0.01, or the mock immunized animals, they all succumb to infection within a week or so. So this gave Ben a really great platform to calculate the correlate of protection against disease or death. And what he found is that there's a beautiful correlation between the amount of antibody against the S1, the pre-infection stage, on the x-axis versus the weight loss at 6 days post-infection on the y-axis. So the more antibody animals generated, the less weight they lost. So based on this calculation, he was able to calculate the amount of IgG required for death and survival, as well as weight loss. And so there's a very nice correlation between the amount of antibody developed, either neutralizing antibodies or anti-spike antibody, and the protection from disease and death. Finally, Ben wanted to understand the role of CD8 T cells in protection against reinfection or mRNA vaccine mediated protection. So he created 2 cohorts of animals, one that have been fully vaccinated with the mRNA vaccine on the top, and the one that has been recovered from prior infection, a convalescent animal. And then he challenged these animals with either the Washington strain of the SARS-CoV-2 or the beta variant. And before doing that, Ben also depleted CD8 T cells from some of these animals. And ultimately, what he found was that all animals that received the vaccine or prior infection were completely protected from death, while all the animals that were not immunized succumbed to infection within 4 days of infection. And when he looked at the viral load, infectious viral titer in the lung of these animals, vaccine and convalescent animals had no detectable infectious titer in 2 days post-infection, whether they were depleted of CD8 T cells or not, and whether they were challenged with the original strain or the beta variant. So this really demonstrated the power of the mRNA vaccine that relies on antibodies, and that the CD8 T cells are dispensable from protection against SARS-CoV-2 infection. So this study illustrated that T and B cells both clear primary infection, that in the absence of one, the other can compensate. However, if you're lacking both T and B cells, animals are unable to control primary infection and they remain chronically infected. Adaptive transfer studies demonstrated the complete protection from serum antibody and partial protection conferred by T cells. And in convalescent animals or in vaccinated animals, CD8 T cells were dispensable for protection against a challenge by ancestral or the beta variant. And finally, he determined the levels of antibodies needed to provide protection based on the mRNA vaccines. So this study illustrates the sort of power and the benefit of the mRNA vaccine in conferring some level of mucosal immunity. So even though mRNA vaccines weren't designed to generate mucosal immunity, we now know from various studies that we can find mucosal antibodies of the IgG and IgA isotypes in the saliva and the nasopharynx of mRNA vaccinated individuals that can have neutralizing activity. That would prevent infection altogether. And even if the virus is able to infect and replicate within the host, there are antibodies and T cell based mechanisms that prevent the spread of that virus. And we already know there's a very effective ability of these mRNA vaccines to prevent symptomatic infections, which then leads to transmission enhancing activities such as sneezing and coughing. And finally, we believe that even transmission may be further prevented by coating of the virus with antibodies generated by the vaccines. So at all levels, vaccines are inducing some level of mucosal immunity due to some crossover from the systemic to the mucosal tissues. However, we believe that the future vaccines, the next generation of vaccines, could benefit from having a mucosal targeting approaches. That's something that we are working on right now. And just finally, I'd like to describe what we are doing right now about the long COVID and why we're interested in this question. So long COVID is happening in millions of people around the globe. That is affecting multiple organ systems that are listed here. These are just some of the over 200 different symptoms that are associated with long COVID. And this is striking in that a respiratory tract viral infection resulting in incredible organ system involvement, including systemic impact, neuropsychiatric, head, eye, ears, nose, and throat, endocrine, cardiovascular, pulmonary, musculoskeletal, and GI tract symptoms. And how can a respiratory virus infection result in involvement of so many organs? Well, one of the hypotheses that we are thinking about is potential establishment of viral reservoir. Now, this can be either infectious viral particle or remnants of virus or just RNA or protein of the virus that's lodged somewhere in the tissue that's not accessible to the nasopharyngeal swabs. And this could trigger chronic inflammation. Another hypothesis we are pursuing is that the COVID infection could trigger autoreactive lymphocytes that target different organs, such as those targeting the antibodies targeting different organs, tissues, and cell types, as well as T cells. There are other hypotheses out there that can explain long COVID, including the tissue damage, dysbiosis of microbiome, as well as latent viral reactivation that's often seen in long COVID patients. So there's evidence for both the viral reservoir and autoantibodies for long COVID. The viral reservoirs have been seen in COVID patients months after the initial infection. This study found, for instance, there are some nucleocapsid staining in the enterocytes of patients that had COVID months before. This is 5 out of 14 patients had positive staining, and 3 out of 14 patients had PCR positive for viral RNA after 3 months in the intestine. There's also evidence of autoantibodies that develop in COVID-19. This is during the acute phase of COVID-19. We collaborated with Aaron Ring, who is a professor in our department where Ting used to work. And Eric Wang and Yili Dai are trainees from his lab. Tiangyang Mao and John Klein are students in my lab. They collaborated to look for autoantibodies in acute COVID patients. This was enabled by Aaron's new technology called rapid extracellular antigen profiling, which enables comprehensive and high throughput screening for autoantibodies based on yeast display library, where human exoproteome are expressed by these yeast cells. And you simply have to incubate the patient's antibody with these yeast cells, and you pull it down using magnetic beads of the yeast that are bound by the IgG. And then you can decode the antigen using deep sequencing. And this enabled us to detect numerous autoantibodies that are found in COVID patients against both immune cells, immune factors, cytokines, interferons, as well as autoantibodies against target tissues. So this is just demonstrating some of the different tissues and different autoantigens that are indicated in the in different rows. And each patient is a column. And patients are divided into severe, moderate, mild, or asymptomatic. And the brighter the yellow color, the more autoantibody there is against that particular autoantigen. So you can see that most patients have multiple autoreactivity against different antigens. There is no unique autoantigen that's found across these patients. However, there are some common ones that are found. And the striking thing is that they are antigens expressed in the brain, vasculature, connective tissue, cardiac, hepatic tissue, saliva, skin, GI tract, and many others. So this illustrates the possibility that COVID-19 can lead to autoantibody induction, which then targets different tissues in different antigens within different tissues to trigger pathological outcome. And there are other reports demonstrating autoantibodies in both acute and long COVID, including the G protein coupled receptors that are already known to elicit a lot of the pathologies. So currently we are working with a number of teams to understand immunological and physiological determinants of disease pathogenesis in long COVID. And this is the only way to inform rational diagnosis and therapy for the millions that are suffering from the consequences. And this was generously supported by HHMI. So I want to end by thanking and acknowledging the authors of the 3 studies that I was able to describe to you today. And I already highlighted the first authors of each paper. And just want to end by thanking the amazing lab members that I have the privilege of working with on a daily basis to try to figure out COVID-19 immunity and to try to dissect long COVID so that we can eventually help these patients in the near future. So thank you for your attention.
All right, thank you, Akiko, for covering so many important topics in the COVID-19 patient immune response. Now we have like 7 minutes for the questions. Okay, Dr. Yang Xinfu.
Hey, Akiko. I have a question related to that two patients that failed to respond to have a neutralized antibody. I wonder whether it's because they lack of a T cell response to RBD, or is lack of B cell response to RBD. Do you have any data for that?
Yeah, so what's intriguing about these two individuals is that they don't have any, as far as we can tell, they don't have any immunodeficiencies. They do develop ELISA detectable anti-spike and anti-RBD IgG, and yet they don't have the capacity to neutralize the virus. So I think the virus neutralization assay that we're using is using the authentic variants, and it's quite stringent. You know, the fact that they can't neutralize it, functionally these anti-RBD antibodies are not being able to block the virus from infecting the cells. So we haven't cloned these BCRs and try to start dissecting the mechanism, but they do develop ELISA antibodies, and as far as we can tell, they also develop some levels of T cell response. So there's something intrinsically faulty about their the particular BCRs that they're using.
And another question about mRNA vaccine. You mentioned about the IgA. Traditionally, when you do intramuscular injection, IgA is very, very low. But I'm not sure when you mentioned about mRNA vaccine can induce IgA, it's because mRNA vaccine have some unique feature, or the IgA still is very low?
Yes, absolutely. So if you compare it to a convalescent individuals, the IgA level is quite low. You can detect it though, and it doesn't happen with adenoviral vector vaccines. So there's something about the mRNA vaccines that's eliciting low levels of mucosal IgA. And there's some evidence that some of them may even be secretory IgA because they can detect secretory components. But again, the level is much lower than a convalescent person. So this level of sort of crosstalk is there, it's present, but it's not efficient.
Yeah. Dr. Bin Su.
Hello, Kiko. Hi, this is Bin.
Hi, Bing.
How are you? I got a question about this long COVID. You showed that there is viruses in the gut. I just curious, in that situation, the host has any immune response, like antibody or T cell responses to those viruses in the intestine?
Yeah, so I only picked that one figure out of that paper, but the whole paper was about the maturation of antibody response that happened in COVID-infected individuals. And essentially, yeah, these people who have these intestinal, PCR positive or antigen positive biopsies, they also are maturing antibody responses over time. So I think what this means is that even in patients who don't have GI tract symptoms or long COVID, there may be a little bit of depot of antigen that happen in the intestine that may be helping them to develop even more somatically mutated and mature antibody responses. However, we also think there's a flip side to it, which is that in some patients, that is actually acting as a source of chronic inflammation if they're not able to clear, the right amount of pathogen there. So we're still, looking at these two possibilities, and intestine is one of the potential target of reservoir.
Thanks.
Thank you.
Okay, Dr.
Hi, Kiko. Beautiful talk. This is Isaac. I was actually wondering about the last part about what are your thoughts about the effects of COVID on the brain. You've done some work in that area, and do you think the autoantibodies that you found with Aaron are involved, or is it direct viral-induced, injury to the CNS that could be part of long COVID?
Yeah, thank you, Isaac. So there are papers, including ours, that detected infection in the brain, and I think it does happen, especially in some severe cases. We don't currently know if that happens in long COVID patients, and it's a little bit invasive to probe that. However, there are studies, including one from Eric Song and Shelly Farhadian, who looked at the CSF from patients with neurological COVID. And they did find autoantibodies as well as antiviral antibodies in the CSF, suggesting that there may be either local infection and or autoantibody induction within that CNS, and that may be leading to some of the symptoms. So, we haven't ruled in either of these things. I think infection in the brain can happen and does happen. How frequently does that happen in the long COVID is a question we don't currently understand.
Okay, Dr. Hai.
Thank you.
Hi. Hi, Akiko. I'm intrigued by the autoantibodies. You think that's special to the long COVID compared to acute and resolving infection? And what about other type of viral infection? I thought when there is inflammation and viral infection typically are associated with some levels of autoantibodies. So could you comment on the spectrum of antigen or the degree of antibody production? But that's where the specificity is.
Yes, so that's a really important question. REAP was recently developed, so Aaron is actually going backwards, trying to understand whether infection other than COVID might induce similar kinds of autoantibodies. So he's collaborating with Charles Dela Cruz, who's a pulmonologist at Yale, who has an archive sample from flu infection prior to COVID. So that's something that they're going to figure out pretty soon. I suspect that you might see some of these autoantibodies develop. And your first question about whether autoantibody may particularly associate with long COVID, that's something we're actually directly looking at, because we have these patients that we characterized from March, April of 2020, who had these autoantibodies. And now we are surveying these patients that are obviously discharged from the hospital. And the survivors, we are now collecting samples and information from these people to see how many of them actually develop long COVID. And the sad thing is that people with that kind of severe disease, they often suffer from post-ICU syndromes, as well as, upwards of 70-80% of these people still have symptoms way past 6 months of hospital just discharge. So I suspect that a lot of them do have long COVID. And if any autoantibodies are associated with that, that's a critical question that John Klein and others are currently asking.
Due to the time limit, let's stop here. Thank you, Akiko, for this insightful talk. Thank you.
Thank you so much. Thank you.
All right.
Thank you.
Our second speaker in the first session is Dr. Isaac Cho. Isaac is associate professor in the Department of Immunology at Harvard Medical School. He received his undergrad training in biochemistry at Harvard College, and he got his PhD in immunology in Harvard Medical School under the mentorship of Dr. Michael Carroll, working on immune responses in neurodegeneration. He then trained with Dr. Tom Manaitis' lab as a postdoc fellow on ALS. He also did his second postdoc fellowship on the neurobiology of pain at Boston Children's Hospital under Dr. Clifford Wood. He studied his own lab in Harvard Medical School in 2014. So his lab studied a very interesting topic focused on interactions between the nervous system, immune system, and the microbe in the host defense and inflammation. His work has found that the nociceptor neurons directly sense bacteria and immune mediators to produce pain. And also, recent work has also shown the immune modulators also regulate itching during the allergic inflammation. And Dr. Chu has received a lot of rewards, including NIH Director's New Innovator Award, Chan Zuckerberg Initiate Ben Barris Award, and Burroughs Wellcome Fund Investigator in the Pathogenesis of Infection Disease Award. And I think his topic today is nociceptor sensory neurons, regulation of the barrier immunity. Okay, welcome, Dr. Isaac Chu.
Thank you so much, Ting, and it's a pleasure to be here with Westlake University opening. It's an honor to be invited. So I'm going to talk to you today about neurons and immunity, and the topic is how do these neurons called nociceptors regulate barrier host defense? So when we think about the nervous system, we have to realize that every organ and peripheral organ in the body, as well as especially the barrier tissues, such as the gut, the skin, the lungs, and I'll talk to you today about the meninges as well, are densely innervated by sensory and autonomic nerves. And the role of these nerves are to take in stimuli from the environment and also to regulate barrier function. So our lab is really interested in how these nerves crosstalk with microbes as well as with immune cells in these key barriers. So today I'll tell you basically two stories, one on the gut and one on the meninges, which are two key barrier sites. Okay, so the neurons we study are those that mediate pain. Why are we interested in pain? Because actually pain is one of the 4 cardinal signs of inflammation. So it's been linked since 2,000 years ago by Celsus to, in a sense, to the immune response. But it's always been thought of for a long time as a symptom, but now we're realizing that the pain-mediating nerves are actively participating in regulating inflammation. And the neurons that mediate pain are called nociceptors. So this term nociceptor was coined by Charles Sherrington to describe the pain reflex circuit, where he hypothesized there must be mechanisms that the body uses to sense noxious stimuli, and then this is transduced into an unpleasant pain sensation and a withdrawal reflex. Well, the neurons that mediate pain, the nociceptors, reside within the dorsal root ganglia. They take in signals from peripheral barrier tissues and transduce them up to the spinal cord, where this is then processed to the brain as a pain signal. And these neurons are able to detect many different types of noxious stimuli, and their role is to protect us from danger. So these include noxious heat or cold, tissue injury, force, reactive chemicals, as well as potentially microbes and pathogens. And on the one hand, behaviorally, this leads to a withdrawal reflex to protect us from further exposure to these danger. But also these neurons can do something else at the molecular level. So one of the things that we found is that these nerves, like immune cells, can directly sense pathogens, just like the immune system. So this is just an illustration of multiple molecular mechanisms by which nociceptors sense microbes. They're able to detect pore-forming molecules from Strep pyogenes and Staph aureus, such as streptolysin S and hemolysins, and this induces pain production. Also, gram-negative pathogens can activate nociceptors through LPS or flagellin via TLR4 and TLR5, as well as TRP channels like TRPA1 and TRPV1. And also, beyond bacteria, Candida has also been found to directly activate nociceptors to reduce pain. So in a sense, these nerves can act like sentinels for pathogen invasion. And then as a consequence, they also signal locally to the immune system. So what's called an axon reflex, where an action potential is transduced along neighboring branches of the nerve fiber that leads to release of neurotransmitters and neuropeptides that then also signal to the immune system or the vasculature. So these neurons are known to immediately induce vasodilation, permeability, as well as signal to the immune system to modulate chemotaxis and priming. one of the key ways that this occurs is through neuropeptides. So neuropeptides, including substance P, CGRP, and VIP, are stored in the peripheral terminals of these nociceptors. And when calcium comes in, they are released, and then these can act on immune cells that have receptors, cognate receptors for neuropeptides, such as mast cells, which can detect substance P via this GPCR called MRGB2. This induces a very significant mast cell activation, so this nerve mast cell axis. Also, CGRP is a neuropeptide that I'll get into that can act on multiple types of immune cells, including dendritic cells to induce IL-23. And also VIP, another neuropeptide, can act on ILC2s. So this is a growing list of interactions between neuropeptides and immune cells that is of interest. And in the big picture, then these nociceptor immune interactions via neuropeptides and other mechanisms are key for barrier immunity. So this is a series of papers that have been shown from our lab and others that neurons in the skin as well as in the respiratory tract regulate the outcome of infection, as well as autoimmunity and asthma. This is just one of the studies we had published recently showing that in skin infections, TRPV1 expressing nociceptors release CGRP, which then suppresses neutrophil recruitment, and that this facilitates survival of a bacteria called Strep pyogenes. And we can block that by having antagonists against CGRP and a release of CGRP that can improve the outcome of infection. So I'm going to tell you today about the gut and the meninges, which are two key barrier sites of our body and how nociceptors regulate them. So the first part is on Nikki Lai's work, who is a graduate student who is working on how nociceptors regulate intestinal host defense against salmonella. So salmonella is a major gram-negative bacterial pathogen that causes 1/4 of all diarrheal disease. And it invades the small intestine of the gut, transiently colonizing the ileum, and then invading via Peyer's patches, which are specialized lymphoid organs in the small intestine ileum. And they invade via these M cells, which are specialized epithelial cells. Upon translocation, then macrophages take them out of the gut and they spread systemically. So we want to ask, do neurons play a role in host defense against this bug? And to study nociceptors, we used different ion channels to target them. So one of the key ones we use to target nociceptors is TRPV1, which is a large pore cation channel that has been shown to detect capsaicin, which is the active ingredient in chili peppers, and heat, noxious heat, as well as low pH. TRPV1 is expressed in the large proportion of gut innervating nociceptors. And just of note, it recently received recognition that David Julius, who discovered TRPV1, won the Nobel Prize this year in physiology and medicine. So how do we target these neurons? Well, we use a TRPV1 DTR mouse, so TRPV1 expressing human diphtheria toxin receptor, and then we injected these mice with DT to kill off TRPV1 expressing neurons, and then infected these orally with salmonella. And what you could see here is that these mice lacking TRPV1 fibers did significantly worse in salmonella infection. So they had more bacterial load in the ileum Peyer's patch in the early time points of infection compared to vehicle-treated controls, as well as spread to the liver by day 5 post-infection. As a second parallel strategy to target nociceptors, we used another ion channel, sodium channel NaV1.8, which is important for action potential generation in nociceptors, and it's not expressed in non-neuronal cells. So here we have a NaV1.8 Cree that we crossed with a ROSA 26 DTA mouse. So this will ablate NaV1.8 lineage nociceptors from development, and Cree negative mice are wild type controls. So we infected these orally with salmonella and then looked at bacterial load at day 1 and day 5 in the small intestine and liver. And what you could see is that this phenocopies the TRPV1 neuron ablated mice, where significantly more bacteria was recovered from the ileum and Peyer's patch at day 1 and also spread to the liver by day 5. So these neuron deficient mice are deficient in host defense. And turns out these neurons, the TRPV1 lineage neurons, which reside in the dorsal root ganglia, can directly sense salmonella. So here, what we're doing is growing dorsal ganglia neurons in culture and stimulating them with the bacteria or with vehicle control. And you could see that subsets of these neurons were directly activated by the bacteria or products in the bacteria. And they also respond to capsaicin, so indicating they express TRPV1. So the neurons are able to sense the bacteria. And I'm summarizing a lot of data. It turns out that the way that these nerves then signal to the gut is through these Peyer's patch M cells. And M cells are these key epithelial cells that sample antigens from the lumen of the gut, but are also exploited by the salmonella for entry. So they reside on top of the follicles of the Peyer's patch, and they have these characteristic microfold shapes. That's why they're called M cells. And so the nerves are actually directly juxtaposed to these M cells underneath the dome of the Peyer's patch. Here's an image of this. You can see these mature M cells marked with the protein GP2. And Salmonella utilizes GP2 as its binding protein on the M cells, and then they translocate through the M cells. So what we found is that these nerves actually regulate the homeostasis of the M cells. So when you target nociceptors, either by using NaV1.8 DTA or TRPV1 DTR, this leads to increased numbers of M cells. So remember, the epithelium is always turning over. The neurons suppress the differentiation and turnover of the M cells. So you can see a baseline, there are more M cells per follicle in the absence of nociceptors. And what we found was that the way that the regulation worked was through this neuropeptide called CGRP. So CGRP actually suppressed the numbers of M cells along the dome of the Peyer's patch. This in turn was also quite important, not only from preventing entry of bacteria which use M cells, but also by suppressing M cells, it allowed a better microbiome homeostasis. So SFB, which are key bacteria that induce Th17 cells, but also protect against enteric pathogens, are able to attach to enterocytes, but not well to M cells. So by decreasing the number of M cells, there's also more SFB, which then protects against Salmonella. So overall, this shows that nociceptors in the gut maintain the homeostasis of the epithelial layer as well as the microbiome to protect against the pathogen. Okay, so this is in the GI tract. And I'm going to tell you some unpublished work that led by two very talented postdocs in the lab, Felipe Ribeiro and Li-Wen Deng. And this is in the meninges. So the meninges I consider also a key barrier site because it is basically the barrier of the brain. And I think you might hear some of more talk in this meeting from Dr. Yoni Kipnis, who is one of the leaders in understanding meningeal immunity. The meninges is made up of three layers, the pia, arachnoid, and dura mater, and it basically wraps around the brain, and it's key for protecting the brain from invasion as well as from trauma. The outermost layer, the dura, is actually quite different than the brain itself in that it lacks a blood-brain barrier, and it actually has fenestrated blood vessels as well as lymphatic vessels, which has been shown by Yoni's lab to drain antigen from the brain, as well as populated densely with the immune cells. So it's a very interesting barrier site for immunity, but also something that maybe people don't appreciate as well is it's also densely innervated by pain fibers. So people in the pain field know this well because these nerves are what thought to mediate headache. So these are some images we took with two different strains of mice in the lab with a NAV-CRE DD tomato, which will label nociceptors that are NAV1.8 positive, as well as calc-A GFP DTR, which labels CGRP expressing nerves. And you can see this is in the dura. So this beautiful innervation pattern of the dura by pain fibers or nociceptive neurons. So also, like I mentioned, the meninges is densely populated by immune cells. So in the brain itself, there's a lot of microglia, but in the dura, there's a whole set of, both myeloid as well as adaptive immune cells and lymphocytes. So you can see these are just some recent studies that looked at the dynamics of immune cells at these border sites. And so actually Yoni's lab and also Marco Colonna's lab recently found that the skull bone marrow also circulates cells back and forth from the bone marrow to the meninges. So it's a really interesting dynamic area full of immune cells. And our question is, do nerves play a role in regulating the immune response here? And specifically in the context of bacterial meningitis. So as the term implies, it is bacterial invasion of the meninges, which affects a large proportion of people a year. And it mainly occurs by spread from the hematogenous route, so from the blood via to the meninges, and then from the meninges into the brain. And mortality can still be quite high. So bacterial meningitis, even if you survive it, there's often post-infectious neurological sequelae. So that includes cognitive issues as well as chronic headache and hearing loss. So understanding how meningitis occurs and also how the host responds against meningitis is quite important. So in order to study meningitis in the lab, we set up two mouse models where we used two different bugs, group B strep or strep agalactiae, which is the leading cause of neonatal meningitis in humans, as well as strep pneumoniae, which is one of the leading causes of adult meningitis in the elderly, as well as in children. So what we do is we infect mice IV with different doses of the strep agalactiae or strep pneumoniae. At a high dose, this will cause, you can see sequential invasion of the dura first, which is outermost layer, and arachnoid, and then the pia and chorioplexus, followed by the brain. So we can study this is bacterial load recovery from each of these tissues. We can study whether nociceptors are involved in this infection. So what we did is we used the NAV 1.8 Cre DTA mice, which have targeted ablation of nociceptive lineage neurons, and the Cre positive mice lack nociceptors and the Cre negative mice have nociceptors. And then we did infection, in this case with strep pneumoniae. And interestingly, we actually found decreased bacterial load in the dura as well as in the brain, but no effect on the bacteremia in the blood. So nociceptor absence actually protects against bacterial invasion. And this was accompanied by significantly more influx of neutrophils and monocytes, which are important in clearance of the bugs. I do want to point out that one of the key mediators of nociception in the meninges is CGRP. So this neuropeptide I mentioned earlier, it's released into the meningeal, into the dura, where it's thought to act on the vasculature as well as potentially on immune cells in headache and migraine. So there's a lot of drugs that target CGRP right now to treat migraine. So we wanted to ask, does CGRP also play a role in meningitis? So in order to ask that, we used CGRP knockout mice. So CGRP has two isoforms, calc A and calc B. Calc A is the major one expressed by nociceptors. And what you see here is infection with calc A knockouts versus wild type or hets, but there's significantly less bacteria than is able to invade the arachnoid or the brain in the CGRP deficient mice. By contrast, if we inject the mice systemically with CGRP in a dose that phenocopies migraine models, this actually potentiates invasion. So you get worse invasion of bacteria into the dura, subdural, chorioplexus, and brain, but no effect on bacteremia. So CGRP is negatively regulating the outcome of infection. And we also found that CGRP is released during meningitis by nociceptors. So what we could do, what Felipe did here was he took a skull meninges explant at 24 hours post-infection versus uninfected mice, and he could detect more CGRP there. And this is a time course of that. And then if we remove nociceptors using NAV 1.8 DTA mice, this CGRP level was significantly less. So nociceptors are releasing this neuropeptide. And also we could find bacteria sometimes juxtaposed to CGRP expressing nerves in the dura. So this is imaging of the bacteria in red and CGRP fibers in green. So could the bacteria be activating these nerves to produce CGRP? So that's what we asked here. So Felipe took trigeminal ganglion neurons, which are the ones that innervate the meninges, cultured them, and then added strep pneumoniae to these nerves. And what you could see is in calcium imaging a subset of nerves that it responded directly to the bacteria as well as the capsaicin, and also that in vitro this induces CGRP release. So we think one of the mechanisms that this is happening is through a pore-forming toxin called pneumolysin. So the Strep pneumoniae uses this to facilitate invasion, but also we think it could activate nerves, because here when we use a Strep pneumoniae strain that lacked pneumolysin, there was less neuronal responses to the Strep pneumoniae. Okay, so the CGRP is being released by nerves. What is it doing? So in order to understand how it's regulating the immune response, we did single-cell RNA-seq of the meninges before and after meningitis, which actually hasn't really been done in the bacterial context. So as you can see here, looking at all immune cells, CD45 positive cells, there's dramatic changes in many immune populations before and after meningitis, particularly in myeloid cells like macrophages and neutrophils. And so in particular, we found that macrophages, here labeled in blue by CX3CR1, are closely juxtaposed to meningeal CGRP nerves. And we could see them in this image. And also many of these macrophages, which also express CD206, increase and respond during meningitis. So could the CGRP be regulating macrophages? So in order to study that, we wanted to first see where's the receptor for CGRP expressed. So the CGRP signals via a co-receptor formed by RAMP1 and calc-RL, which is a G protein coupled receptor, and this induces cyclic AMP signaling in immune cells. So looking across the single cell data set, we could see that calc-RL and RAMP1, but actually not so much these other neuropeptide receptors, is highly expressed actually in many, many immune cell types, including macrophages and neutrophils in the meninges before and after infection. And to target it, what we did is we first crossed the RAMP1 floxed animal with the lysozyme Cre. So this allows elimination of RAMP1 in most myeloid cells, including neutrophils, monocytes, and macrophages. And upon infection of these mice, what you can see is lack of RAMP1, the receptor for CGRP, improved the outcome of infection. So there's less bacteria in the dura and the brain, but not effect on the bacteremia. But lysozyme does not specifically target macrophages. It's also in neutrophils. So in order to go after macrophages only, we looked for a specific macrophage marker. So looking at the single cell data set and also looking at some published papers, one of them, PF4, is actually expressed in macrophages at pretty high levels, but not in microglia in the brain. So this is actually a PF4 Cre tomato mouse. So you can see that these meningeal macrophages are labeled and they're lining the blood vessels. So we crossed the PF4 Cre with the RAMP1 flox mouse, and this allowed us to specifically ablate RAMP1 in macrophages. And this also showed significantly worse, so a better improvement of infection with less bacterial load at 24 hours in the brain and dura. So we think macrophage signaling of CGRP is important. And so how does this occur? So what Felipe did was he injected CGRP into mice and did single-cell RNA-seq of mice with and without CGRP after meningitis and compared the immune response. And what he found was that in macrophages in particular, there's a big polarization of the immune response where CGRP would turn on many genes on one side, but also turn off some potentially key genes such as chemokines. And so we think the chemokines such as, here CCL2 and CCL3, which we looked at in vitro, are decreased by CGRP. And also TNF alpha, which is of course a key pro-inflammatory cytokine that could be involved in host defense. It did not seem to affect macrophage killing of the bacteria. So this suggests that macrophages are being dysregulated by CGRP. And this is kind of confirmed here where in the RAMP1 knockout mice, we also see basically that, you get more monocytes and neutrophils, which we think are recruited by the macrophages via their chemokines and cytokines. So this also begs the question, if RAMP1 or CGRP blockade is so effective, potentially this could be used as a treatment for meningitis. And since there are anti-RAMP1 treatments right now for migraine, we tried using that in our model. So here we use a RAMP1 antagonist called BIBN. And what you can see here is that treating mice with the BIBN during infection can significantly decrease bacterial invasion of the dura and the brain for both strep Echolactia and strep pneumoniae. So what I've shown you here in the second part of my talk is that in the meninges, another key barrier site, these nociceptors are impacting the immune response. So actually, the trigeminal neurons are responding to the presence of strep pneumoniae or strep Echolactii to secrete CGRP. CGRP, on the one hand, probably drives significant pain and headache, which is a characteristic of meningitis, but also signals to resident macrophages there, which then regulates or polarizes the macrophage response, which then inhibits antimicrobial defenses. And this may be exploited by the bacteria. For example, the bacteria can activate the neurons via pneumolysin. So if we block this pathway, we can enhance immunity against the bacteria. So in summary, what I've shown you is that at two key barrier sites, nociceptors and immune or nociceptor epithelial interactions mediate antimicrobial host defenses. In the gut, TRPV1 neurons regulate the homeostasis of Peyer's patch M cells, and these are key entry points of salmonella, so this protects against salmonella infection. And in the meninges, the nociceptor signal via CGRP RAMP1 axis to macrophages in streptococcal bacterial meningitis. So really, these are just two cases, but we think that understanding how neurons crosstalk with immune cells at these barriers could lead to new treatments for infection and inflammation. And this was all work done by very talented people in the lab. I mentioned Nikki Lai, who did the work on salmonella, and Felipe and Li Wen, who did the work on meningitis. And these are some really great people, members of my lab. And we have key collaborators, including Mina Rao, Jun Hu, Christophe, and Diane on the gut work, as well as Kelly Duran, Dan Levy, Rick Malley on the meningitis work. So, and very generous funding sources. Happy to take questions.
Thank you, Isaac, for this wonderful talk on how these nociceptor neuroimmune interaction can do both antimicrobial, also do protection role in the meninges. Now we have 5 minutes for the question. Yeah, Dang Yang. Oh, Dan Shen, you can go first.
Hi. A couple of questions. First, this nociceptor regulation of immune function. Is it neuronal autonomous function, or do you think it involves circuitry, neuronal circuitry?
It's a good question. So I think it will, depending on the situation. So the work that I've shown is really autonomous. So it's happening locally because of the neuropeptide release. But these neurons will also induce central circuits like stress signaling, which could, have major, feedback loops via the brain to the immune system, such through the HPA axis, for example. So I think that, like we had found actually that pain induces stress, which then actually feeds back. This is in collaboration with Yachi Xu on inducing stem cell loss in the hair follicles. So there's I think there's going to be long distance circuitry induced immune responses and also local nociceptor immune responses.
Okay. The second question, that in the case of a meningeal infection, the CGRP released by the nerve actually inhibits antibacterial defense. So why do we want to do that, and what's the function of that mechanism? Right.
It's a great question. It's puzzling. So I think that possibly it's protective in other cases, like when you have too much inflammation, right, and potentially in traumatic brain injury, or maybe even other pathogen infections where the immune response, too much of immune response, is actually a bad thing. So I think it may depend on the situation, and we know actually in meningitis is a complicated case where actually you have to treat with steroids as well as antibiotics in patients. So, the immune regulation at that border is a complex area, and I think nerves need to tune that. So, I hope that answers the question.
Yeah, Danyang.
Hey, Isaac, a very excellent talk. I have several questions. So, I wonder if the innervation in the meninges have some functions under even hemostatic condition, like regulating lymphatic drainage, or like attracting immune cells from the skull, or other regulatory functions.
Yeah, that's a very good question. So as I mentioned, the main role of those nociceptors is to mediate pain and headache. So I guess the question is, when do you have a headache? Maybe when you're feeling sick, but also it helps you want to lie down and sleep, and maybe that helps with the lymphatic drainage point. But like your point is, are there other roles of these in maintaining the homeostasis of the meninges? So that includes potentially regulating lymphatic function and vascular function, as well as the immune homeostasis at that border. So I think that's probably going to be happening.
Thanks. I have a question related to the first part. So there are CGRP positive neurons in the gut as well, intrinsic innervation from the enteric nervous system, right? I just wonder if this CGRP neurons also contribute to bacterial infection in the GI tract.
Yeah, that's a great question. So what you're referring to, there's of course the enteric nervous system, and there are sensory neurons there that express CGRP. We kind of ruled them out in a way because most of those express CGRP beta. And when we looked at CGRP beta knockout mice, there was no effect on the infection, whereas CGRP alpha, which is mostly in the extrinsic nerves, play the key role. But I think that in other, I think we have to think about, the gut is probably the most complex in terms of sensory innervation. So, the different parts of the gut, the small intestine versus the colon, there could be different roles of intrinsic versus extrinsic sensory neurons, and the role of CGRP source from each of these could play different roles in each situation, different depending on the pathogen, the type of inflammation.
Thank you.
Okay, Zixu.
Yeah, I mean, it's a wonderful talk. I mean, I like it. Talk very well. So I have like a really quick question. So you show like depletion of the TRPV1 and also level 18. So you show the M cells were increased. So I was wondering if this really happened during the Salmonella infection. And the second thing is if this apply to the other like bacterial pathogens, since a lot of bacterial pathogens they use M cells to get into the gut, right? Thank you.
Right. So actually, you're right. Actually, a lot of the data I showed was actually at homeostasis. So meaning that this is happening even before infection. But we also saw after infection, the numbers, so M cells are actually increased in number by Salmonella. So they will induce M cell differentiation. So nociceptor deficient might have even more above the increase that you see. So I think it's regulating when you have homeostasis, but I think that the mice will often be exposed to things that can activate these nerves. So it could be something in the diet, like I mentioned capsaicin. It could be something in the microbiome. So we're thinking the microbiome actually regulates nociceptors at small levels at homeostasis. So kind of what activates the nerves at homeostasis will maintain some low levels of regulation. And then when you have a major damage, which could be a pathogen or it could be something noxious in the gut, then you have a much higher level of regulation.
Yeah, Dr. Haiqi.
Hi, Zach. I just have a quick question. I want to clarify whether I misunderstood. You mean the TRPV1 neurons directly sense the bacteria-derived product in the gut lumen, then interact with the M cells to effectuate the increased number. Is that correct, or?
I think it's theoretical, but I think that the nerves can be can be activated via multiple pathways in vivo. So in vitro, we know it can respond to Salmonella, but whether that's the actual trigger in vivo, we don't know for sure. So. It's right. So I think that's fair, very fair. So I think, enteroendocrine cells are really key in the gut. And those are, in normal situation when there's no barrier compromise, they're the first ones sensing luminal contents. And they actually have some interesting nociceptive characteristics. They have TRPA1, which is another nociceptor ion channel, and they have receptors for short-chain fatty acids. So they could be the ones first talking to these nerves via serotonin. But I think I guess our argument is during barrier compromise, then the microbiome or, salmonella and things can directly talk to the neurons. And I think generally the nociceptors are just tuned to anything that's very tissue damaging. So it could be heat, it could be, noxious things from bacteria, it could be, I guess, also spices which mimic those things. So, yeah.
Liang, for the last question.
Hello, Isaac. Yeah, nice to see you. And I have two questions. The first question is, as you mentioned, the bacteria like Streptococcus pneumoniae activate neurons by agents like pneumolizing. I wonder whether this interaction is specific to the nociceptor expressed in neurons or sensory neurons. This is the first question.
That's a great question. I'll have to ask Philippe. So this is some of the data is a little bit preliminary, so we're still analyzing it. What we found with other pore-forming toxins like alpha hemolysin or the strep-derived streptolysin S is at low doses, it seems to be specific for nociceptors. But when you get higher to the point where it's very lytic, then every neuron will respond. So it's not, so there might be something about the nociceptor membrane that's a little bit more sensitive to low doses of the toxins. But yeah, once it gets higher, it's no longer so specific. But I also feel like it's kind of these things where these are noxious, highly damaging molecules. So it may make sense that a pain fiber, would be able to respond, right? When you need it to respond.
Question is rather quick. So, is there any common ways that bacteria can actually fight back to block the neuron sensing mechanism that can benefit to their infections?
Yeah, no, that's a great question. So we have we're very interested in this. So there have been some analgesic bacterial products that have been identified. So like certain probiotics, people are trying to find things that block pain. So there was a lipoprotein called E. coli that was found to block pain. There are some bugs that may block pain to help it, spread. So we actually have a study coming out on anthrax toxin, which we found blocks pain. And we know it's actually causing painless lesions. So maybe in the skin, this will facilitate its spread, from person to person. This is a fascinating area. So bacteria may exploit nociceptors also to activate them. Like there was a nice Cell paper from Mike Shiloh's lab showing that tuberculosis activates nociceptors to induce cough. So that could help it spread. So the whole dynamic of how bacteria target neurons is very interesting.
I think time is up. Thank you, Isaac, for this wonderful talk. And
thank you.
Okay, our first session in the morning now ends. Now we'll have a tea break. I want to highlight that I would like to encourage all the trainees to ask questions. This talk is a good training opportunity for you to engage with these international speakers or so. So I would like you to bravely ask a question. All questions are welcome. Yeah. I hope you guys can have more questions later. All right. Thank you.
Hello everyone. Welcome to the second session of this meeting. In this section, we will have three outstanding speakers focusing on different aspects of human immunology. So please allow me to firstly introduce our first speaker, Dr. Ren Sun, who currently is a professor in the University of Hong Kong. Before joining the University of Hong Kong, Dr. Sun was a distinguished professor of pharmacology of the School of Medicine, as well as bioengineering of the School of Engineering at UCLA. Dr. Sun's research really is trying to apply systems biology approaches to address critical viral and host interactions. So very recently, his lab has established a platform to profile antibody response at a single amino acid resolution, as well as at a very large genomic scale, which I think he will talk about today. So let's warmly welcome Professor Sun for his talk.
Hello? Hello, can you hear me? Hello? Okay. So first of all, thanks organizer for offering this opportunity to share my experience. Today I will describe the new technology platform established at Hong Kong. It's a transition for me to be from biologist to try to learn immunology. So in the past 30 years, I have been working on virus and looking at how virus and leaving US and move back to China, give me opportunity and also out of necessity to start a new research direction to not overlap with the research has been done in the US. So we are starting looking at it, how the immune system interacting with the pathogen. And so I will share the experience, how we move from being. So first couple of minutes, I'll describe our biology experience and how does that lead us to immunology. Several years ago, we started to have a systematic exam of the virus genome, and then we mutate every new one mutation at a time across the genome. And we pulled our mutants together and look at the phenotype of all variants. So here, and the length of the bar reflects how well the virus replicates in different conditions. So this is a functional genetic map of the virus genome. And from that functional map at the different conditions, this on this side is the virus replication. Okay. On this side is how virus replicates without adding interferon. And on the right side, my right side, and look at how virus replicates in the presence of interferon. So at the different selection conditions, how each mutant behave, and then the difference between those two conditions reflects how virus interacting with the immune system, with the interferon. And from this, when they mutated, reduced virus replication in the presence of interferon, but did not reduce the virus replication in the absence of interferon. And we combine 8 mutants together, and each mutant reduced replication, and the replication was reduced as a vaccine. So the virus cannot replicate in normal mice, and the virus replication
So we are encountering some technical problem to bring Sun back. So then we will directly move to the next speaker, and then we will call Professor Sun when he is available. So sorry about that. So our next speaker is Dr. Hai Qi, so who currently is a tenured professor of immunology at Tsinghua University. He's also serving as the Dean of School of Medicine at Tsinghua. So Dr. Qi is really one of the international leading scientists in the field of germ center B cell selection and humoral immunology. So original work published from his lab in roughly the last 10 years has really been helped us to understand how high affinity antibodies are generated at a molecular, cellular, and systems levels. So his work has been recognized by many significant international and national awards. So for trainers, I want to mention that Dr. Qi's talk is always very insightful and beautiful. So I hope you will enjoy it. So let's welcome Dr. Qi.
Thanks for that nice introduction. That's put the extra pressure on me. So as Heping mentioned, that thanks to his graduate work that we have been studying terminal centers for a while, and we try to understand how this remarkable dynamic structure organized and function to produce the vaccine response that we have heard in the first session. And also, the potentially dangerous response from the autoimmune response. Okay, yeah. So the title is two parts. It's a selection and output, but then I decided I will focus on the selection. But the reason that we study the selection is really because of the, why is it, I don't want to, okay, let me control that. I can control that. So this is the place where our immune response generate, it's just a pointer, it doesn't work. Oh, here, okay. From here that you output this after selection, memory B cells or plasma cells that underlie our useful immune response, but sometimes when it goes wrong, it can also lead to autoimmunity and lymphoma. The reason behind that is because in order to generate the high affinity antibody, the B cells here need to hypermutate. They have to generate a secondary diversity of their BCR, and that's being selected based on their binding affinity to antigens through a process that's still incompletely understood. And that's what I'm going to tell you about today. So we know B cells produce the antibody, but B cell doesn't just become good plasma cells on their own. It needs to talk to many other cells, and one of the most important one is the helper T cells, that in a nutshell, the B cells need to acquire antigen based on their binding affinity, then present it to the peptide MHC to the T cells that recognize that through their T cell receptor. And T cell will give the B cells a variety of helper signals. Among them, one of the most important is CD40 ligand, that really, it's one of TNF family cytokines that membrane bound, but tell the B cell to survive or proliferate and do a variety of things that are necessary to become a good plasma cell. So this information exchange, of course, is context dependent and require time. So the two cells have to come together and stay together for a reasonable amount of time in order for the information to transmit. But if you look at the dynamic structure here, the red cells is the germ center cells and germ center B cells, and the blue and the green cells are the T cells. They don't necessarily stay together for a very long time, even though that I have accelerated the movies for 300 times. On average, they actually stay together for 3 to 5 minutes. So it's hard to imagine that it would finish this information exchange within such a short period of time. So in essence, the B cell need to sequentially talk to different T cells. And so that brings an interesting thought experiment, that how do the B cells decide to to do what they are supposed to do after the selection. So I will list it here. So even though we don't know the molecular details, one can assume the B cell need to have a signaling integrator inside the, be it epigenetic, be it the signaling pathways, something that need to account for how much help signal they receive from the T cells. And based on the experiment that from many different groups, we know that the B cell will die if it doesn't get any T cell help inside the germinal center. Then we call that a selection failure. But hopefully before they die, they receive some T cell help that I will use an arrowhead as a thought experiment as an indicator. If it receive, then I will rescue the B cell from the death by neglect. But if it continues to receive multiple delivery of help, then hopefully they reach accumulate the signal to a certain point that it will pass a selection threshold and become successfully positively selected. Then the B cells can go on to do, either stay inside the germinal center to amplify the response or go out to become memory cells or plasma cells. And that decision point, how did the B cell make that decision, is really the topic of entire different talk that is still being actively pursued by many different labs in the world. Today, I'll focus on this question is, before that, what if we miss one of, so that the B cell do not get a frequent T cell help, then what happens is the B cell would presumably will oscillate within this two threshold is until either die or they're being positively selected. So the essence of being positive or successfully selected, it seems to be, given the dynamic nature of this, the germinal center, is that the B cells need to productively interact with the multiple T cells at a sufficient frequency. If we can sort of translate have this scientific question. Now, how that is being managed, you can imagine in this dynamic context of dynamic interaction, everything can happen in random. So some B cells will just randomly bump into different helper T cells in a productive way, then they will acquire sufficient help to be positive selected. That still can work. But we wonder whether that can be facilitated in any way. And I think that one of the interesting features of the immune system, when we get to the level in the single cell dynamics, it's the stochasticity and the deterministic, it's always a two side that one has to consider. And if there is something that can facilitate the process, it typically can always, you can find evidence for that. And that's what we are looking for. We want to ask whether T cells really can tell good B cells, even from a distance. That would make a contact-dependent B cell help more efficient. If that's the case, then we assume that there should be a process that for B cell to attract the T cells according to their affinity. So the point is you want to select the high affinity B cells, and whether naturally rising high affinity B cell would have a better ability to attract the T cell help, then they can focus on to themselves to receive multiple T cell help within reasonable period of time to be selected. But with this sort of idea, I think for the audience, the students sitting in the audience, you probably scratch their head. Do you want to really go on this potentially goose hunt to find evidence for that? And it's actually not easy to convince students to do that. And I wasn't, at the beginning, wasn't brave enough to look for the evidence until something that showed up actually by sort of an accident. So when we were studying a transcription factor BCL6 in the T cells. So I think for some of you, you will be very familiar. This is considered a master regulator for helper T cell inside the germinal center. If you knock it out, then you lose the germinal center T cells and you lose the germinal center itself. But when we have a heterozygous BCL6 mutant in the T cells, the TFH cells or the helper T cell inside GC look normal, but they show a very obvious defect in terms of their calcium signaling beneath the TCR stimulation. And when we were imaging these cells, we realized an interesting phenomenon. I will show you this movie where the B cell is green and the knockout cells, or heterozygous knockout, is green, red, and the wild type is blue. And we will watch how these two cells chasing the green B cells. This is B cells will highlight. Whenever there's a T cell bump into it, it will highlight with the white line. So what you realize is that this wild type T cell seems to be able to bump into the T cells, B cells more frequent, or the other way around is the B cell seems to attract the wild type T cells better than the heterozygous knockout can sense that B cells. So at the time we realized that this dynamic feature suggests something that controlled by B cell 6 is impacting on how T cell perceive the B cells. And eventually we realized B cell 6 at least in part control the calcium signaling inside the T cell, be it triggered by the TCR or be it triggered by GPCR. So, but that gave us a more concrete evidence, say, this almost certainly is a chemo-attracted process. It's something that's different between these two types of cells in a calcium-dependent way that's sensing the B cell. But then there has to be a chemo-attracted receptor and ligand involved. And that gave us a more concrete hypothesis to look for. And so a former graduate student, Liu Bo, when he did rotation in the lab, he started this heroic effort that we start to screen for chemokines expressed by germ center B cell. And the experiment that we set up, now looking back, it's really important to use the CD40 stimulation that I will explain toward the end. So basically, germ center B cells, in vivo, we briefly stimulate with agonistic CD40 antibody to mimic the T cell help for a very short period of time, then look for all the chemokines that potentially be expressed, and we realized that there's one chemokine, CCL22, that is dramatically upregulated by more than 1,000-fold. And we know CCL22 act on CCR4 receptor that has known been expressed by the T cell. That gave us potentially a ligand and receptor pair that would regulate the germ center B cell attraction of T cells. And so that's our hypothesis to test. So of course we want to verify the T cell express CCR4 receptor, so we made a CCR4 knockout as a negative control. So helper T cells express CCR4, regulatory cells as previously have been found that they express high level of CCR4. The regulatory T cells inside the follicle, we call the TFR cells, also express CCR4 similar to TFH. And this is just a quantitation saying that T cells do express, and as expected, a conventional Treg, for lack of no better words, a conventional Treg, express the highest level. And functionally, the Treg cells are attracted to the chemokine better than everybody else. But within the follicular context, the TFH cells respond to CCL22. It has a cousin chemokine working on the same receptor called CCL17. So we can here. Then the TFR cells do not respond functionally to the chemokine. So that creates a situation inside the follicle. The chemokine potentially would only attract or more strongly the helper T cell. We don't understand fully why regulatory cells do not respond chemokine when they express the chemokine receptor, but like many other things, the Treg cells have the apparatus, but they respond differently than the conventional T cells. So that's an interesting topic, but that's aside. So with this chemokine receptor expression, now we want to understand inside the germ center whether there's really evidence for B cell to attract the T cells using this receptor. So because we had the previous finding with the BCL6 heterozygous knockout, then we set up exactly the same system, but now we are comparing the T cells that lack a CCR4 receptor or express the receptor. And the whole system is, they have antigen specificity, OT2 cells recognize OVA, and MD4 cells recognize HEL, and we can use a model antigen, HEL OVA, to generate the germ center response. What you will see is a focus on particular B cells, and we'll see how these B cells be chased or attracting the two type of T cells. Whenever there is a contact, we'll highlight with the broken white line, and it will, hopefully you can appreciate how the red B cells would differentially attract this two type of T cells. So you can see the cyan colors T cells bump into the red cells are more frequent than the green knockout. Of course, because in order to do this experiment, we always have to do the fluorescent protein transgenic, so it's important to actually verify what we saw isn't because we are using two different fluorescent protein transgenic line. So here is the wild-type comparison using the same CFP-GFP transgenic. Now, if you look at this red cells, you can see there are blue and the green cells bump onto this red cells more in a comparable manner. So of course we have to quantitate, because in all the experiment we have wild-type internal control, and we know the tissue volume we image, we know the number of cells inside the tissue volume, and so we can have a very well controlled quantitation of how frequent they hit each other. And there's a clear, when the T cells lack CCR4, they have a much markedly reduced ability to chase the B cells. And we verified, just to make sure this is not a knockout developmental defect, we also conduct experiment with shRNA knockdown of the mature T cells, and we got recapitulated the same finding that suggests well, rule out the possibility it's a developmentally different in terms of the T cells. And then we verified the CCL22-17 double knockout B cells will have the same phenomenon, show the same phenomenon of differentially attracting T cells, and their contact durations within our imaging period